All right, guys. Thanks for being here. Those of you guys who are on Zoom. I am just going to really quickly double check that we are recording. Mm-mm. Okay, looks like it is recording. Can somebody who is there let me know? Can you just confirm that you can hear me and you can see my screen? All right, awesome. Thanks for the thumbs up, Maddie. All right. So we're going to continue with actually the second lecture file today that we started on Monday, or no, last Friday. So this is the phylogeny development and body plans lecture. Um, Let me go. I'm just going to where I think we stopped last time. All right. I think we were talking about feeding. Is that right? And you guys can ask any questions you have in the chat, or you can use the little hand raise gesture. I should be able to see those, although there's kind of like a lot of things on my screen that I'm trying to look at. All right. So feeding wise, we started discussing this last time. So this is again in the context of circulatory systems and sort of getting our nutrients and gases and things that we need in and out of bodies. So phagocytosis is the basic mechanism that organisms use to engulf and consume food within the cells. So this is what you're going to see mainly in unicellular organisms, but also sponge cells do this very same thing. So essentially, they're just having a little invagination of the plasma membrane of the cell that engulfs the food particle, and then that'll form a vacuole that will join up with a lysosome to digest that food, and then the lysosome will break open and the digested nutrients will then be in the cytoplasm where it can be taken up by different cellular components. Um, so in most animals, however, we have a gut, which is basically an external compartment for digesting food. So technically, the digestion occurs outside of the body. We mentioned that a little bit last time when I compared an animal with a through or complete gut with two openings to a water weenie. Um, yeah. I actually, oh, I meant to add it. I was so amused with myself. I made a little PowerPoint slide last time. I'll try to show you guys that at the end. Anyway, so most animals possess some kind of internal digestive tract. If you have an incomplete gut, you just have a blind end gut sac, essentially, with only one opening. And this is what we see in cnidarians, like our jellyfish and corals, also in flatworms, which are our platyhelminthes. And these organisms basically have to eat their food and then digest it and then spit the waste back out the same way. If you have a complete gut, though, you have a mouth and an anus. I personally am very thankful for that. A famous quote by Libby Hyman, who's an invertebrate zoologist, saying, "The advantages of an anus are obvious." I'm very amused by that. Um, yeah, I'm personally very thankful. But anyway, most animal phyla do have a complete gut. So as we go through and we start talking about the phyla, this will be one of the things, one of the characteristics that we will mention. All right, so excretion and osmoregulation. So one of the issues that we have, of course, is that we need to get waste products out. And then the issue of osmoregulation has to do essentially with ionic concentrations and keeping them where we want them inside of our cells versus outside of our cells. Because, you know, if you are a marine organism, for example, in a lot of cases, you actually need the salinity inside of your body to be lower than the salinity outside in the environment. And so that's an issue. And we face the opposite problem if we are an organism living in freshwater. You need to keep a lot of important ions like the sodium and chloride you need for your muscles to function. You need to keep those concentrations inside of your cells high, even though outside is lower. And diffusion and and also osmosis, the movement of water molecules, is not going to be favorable for that all by itself. So excretion is just eliminating metabolic waste products, everything with a metabolism, and so all animals need to do that. We have nitrogenous wastes that are going to come from the basically breakdown of amino acids, which are your components of proteins. Osmoregulation, as we said, is your regulation of ions inside of your cells. Super, super important. So we need to keep our nutrients inside of our cells, those solutes, for our metabolism. And as we said, we also need certain ions for muscle function and things like that. So yeah, so we already mentioned in a marine environment, a lot of organisms are going to osmoregulate, but a lot of them, and including a lot of invertebrates, actually have adaptations to basically be what are called osmoconformers, where they essentially just, I don't want to say give up, but they just, you know, maintain the same osmolarity as their environment. So if they're salty, they just keep the internal part of their body salty. It does require less energy because osmoregulation is energetically expensive. Um, and so a lot of organisms have to do this in marine environment, and pretty much all freshwater organisms have to be osmoregulators. Because if you live in fresh water with an ionic concentration near zero, obviously if your cells also have an osmotic situation, an ionic concentration of zero, you're going to be in trouble because there's all kinds of stuff we need in our cells to survive. So osmoregulators are going to actively transport things across their membranes. All right. So that's kind of circulation and dealing with waste, and that also has to do with limitations to body size that we talked about last time. Other kind of animal characteristics that we were discussing are support structures. So some of our support structures are made out of that protein that we talked about that animals all have. Does anybody remember the name of that protein? You can answer in the chat if you want, or you can unmute yourself.
Was it collagen?
Collagen, yeah, thanks, Alex. So some of the support structures have collagen as a component, but not all. Obviously, if we have some kind of skeleton, whether it's internal or external, the skeletal structure is just a hard structure that provides support for organs and muscles and things and maintains body shape. Um, Most animals do have some kind of skeletal support. A lot have an exoskeleton, like our arthropods, of course. Organisms can also maintain their body shape in other ways. So some of the inverts that we'll talk about have what we call just a hydrostatic skeleton. And this just means that rather than having like hard structures that maintain their sort of body shape and the position of their organs, they're essentially relying on the pressure of the liquid inside their bodies. Um, so yeah, we have things like roundworms have really high pressure in their bodies. And so the way that they basically maintain that sort of rounded cross section that we'll see when we get to them is by just having a lot of pressure. So they have a hydrostatic skeleton. Um, Organisms that have sort of hydrostatic support can also have circular and or longitudinal muscles present that help maintain body structure. So again, we'll get to that more when we talk about various worms. Segmented worms have both kinds of muscles, circular and longitudinal. That's our annelid worms. Sea anemones also have musculature that help maintain their shape. And then rigid skeletons, of course, that's what you more traditionally would think of as a skeleton. So advantages, we can attach muscles to them, and that essentially allows work to be done like kind of in opposition to those hard structures so that we can move limbs and things like that. Um, Rigid skeletons also can give some support to body shape, especially when you're talking about exoskeletons. Um, they also, because they're providing this structural support and basically working against gravity, rigid skeletons are actually really important for having a larger body size or living on land, where we're not just kind of buoyant in a watery environment. Um, because of the attachment sites for muscles, we can also have precise control of movement. Obviously, hard structures protect us from damage. You can move faster, again, if you have those hard structures that muscles can attach to. So lots of advantages of having a rigid skeleton. And again, we can have an endoskeleton or an ectoskeleton. Developmentally, and this will mean a little more when we start talking about development. Developmentally, the skeleton comes from different tissue layers that form early in embryonic development, depending on whether it's an endo or an exoskeleton. So if you have an endoskeleton like chordates or echinoderms, then that's derived from the mesoderm, kind of that middle embryonic tissue layer. If you have an exoskeleton, it is from the ectoderm. So our arthropods and things like that have this skeleton developing from the outer tissue layer that forms during early embryonic development. All right, any questions so far? Nope, all good. All right, I'll try to remember to pause every once in a while, but again, you can just type in the chat or use the little hand raise function if you have a question. Don't feel like you're interrupting. Okay. I am going kind of fast because we're already behind because we had that whole awesome discussion about what constitutes sexual reproduction last time. All right, so I'm gonna skip over a little bit of this because it's kind of yeah, you already know this. So the exoskeleton of arthropods is kind of interesting. They have chitin, which is actually a kind of carbohydrate, and also proteins that are kind of cross-linked with each other through this process that's called sclerotization, aka tanning. And these can also be mineralized, usually by calcium carbonate, which is what we see in crustaceans that have like a super hard exoskeleton, and also that have access to a ready supply of dissolved calcium carbonate from living in seawater. All right, so that's kind of structural support and stuff. So let's talk about some more of the characteristics of animals. So we're gonna talk more about muscles coming up next, and also nerves. So muscles and nerves. So muscles, what are they? Basically, they are contractile cells that are going to allow animals to move. So muscles can basically shorten and lengthen, whether or not they're attached to some kind of skeletal element or a hard skeletal element varies. Um, so sponges, we talked last time about how they're kind of the problem children in terms of these animal characteristics. But sponges do have these contractile cells that have homologous molecular machinery to muscles and essentially seem like they're kind of like proto muscle cells. So, you know. Nerves. So nerves are basically these bundles of fibers that transmit electrical impulses, essentially. All right, and so when you have muscles and nerves, a really important and big thing that this allows animals to do is locomotion, of course. Um, so pattern-wise, we can have amoeboid locomotion. We're not really going to talk about this because we mostly see it outside of the metazoa. However, we do see it in especially some of the types of cells in our sponges that we're going to get into when we get into the periphery. We also interestingly see this type of amoeboid movement in like some of our white blood cells as chordates. So components of our immune system that have kind of really ancient characteristics. Um, you can also have cilia and flagellae that allow for movement. Nearly every phylum of animals has cilia or flagellae in some kinds of their cells. In some animals, like in comb jellies, the ctenophores, like this guy in the GIF here, the cilia are actually really, really large and also fused together to make these comb rows that allow for sort of the macro movement of the whole animal. And that itself is not very common in a lot of animals. We can also have hydrostatic locomotion. that involves muscles, or we can have locomotor limb movement. Again, that is usually going to involve muscles working against hard, rigid skeletal elements. All right. Collagen, which Alex reminded us, is this protein type that pretty much all animals have. That's really an important component of connective tissue, including your skeletal tissue, including the tissue that gives support to like the walls of your blood vessels and to your skin, and things like that. Um, Collagen is basically like a cable. It's like molecular ropes. So it's literally these proteins that are wrapped around each other. So usually you have these linear collagen molecules. Again, it's a protein, so it's made up of amino acids, and then they are cross-linked with one another. That's what's represented by these little pink guys. And then they twist around each other in a triple helix. And that is what makes them so strong. So it's literally like how the structure of a rope is made. Um, Only animals have this type 4 collagen. Okay, so collagen, that's super, super important. Also, yeah, in our bodies, collagen is usually going to be 30% of the protein, like total protein. So that's a lot. All right, so it's muscles, nerves, and collagen. Um, and now I want to talk about development a little bit. So we're going to talk about yeah, that egg development, and we're also going to talk about embryonic tissue layering. Okay, so here's what happens in animal cells when you have sexual reproduction. You're gonna start out with a fertilized egg. We already talked last time about how our eggs basically are a result of meiosis, but all of the cellular goodies end up in one of those products. So you actually only end up with one egg cell per original parent cell in meiosis, making eggs in animals, only in animals. But sperm, you end up with four daughter cells for every original cell as products of meiosis. So of course, when the egg and the sperm cell come together and you have fertilization, and at that point, the thing that you end up with is known as a zygote. And then animals are going to go through these really characteristic stages of development where the cells are splitting and organizing. So cleavage is the word that we use for that process of cell division. So the cells are basically each going to split in half during each division. So you go from 2 to 4 to 8 to 16 cells and on and on. Eventually, you're going to end up with a hollow ball of cells. So they'll actually have like a space in the middle that's not filled in, and all of the cells are around the outside. This is called the blastula stage. So this is a blastula. And if we look at a cross section of it, that hollow space inside is a blastocele. Okay. That word cele, that suffix, you're gonna see it again. You're gonna see it multiple times. That just refers to a body cavity or some kind of cavity. Okay. So we got the blastula with the blastocele. And then the next stage is where you're going to start to form the gut. And what happens is this invagination of that blastula. And that at that stage, once we undergo gastrulation, we have a gastrula. Anybody want to remind us or just tell us, what do you guys think or know that that prefix gast or gastro refers to? Yeah, Parker said stomach in the chat. So, yeah, so essentially gastro is referring to the stomach or gut, right? You go to a gastroenterologist for digestive issues. So you can remember that this is the gastrula because we are forming essentially the beginning of what will be the gut in our adult organism. All right, so we've got gastrulation to form the gastrula, which now has an opening. The opening there is called the blastopore. We've got now an area inside that's essentially going to be the lumen of the gut. In this early gastrula stage, that's known as the archenteron. Arc is referring to early, right? Like Archaeopteryx is like an early bird. So that's what arc means. And enteron is another word for a space. I don't know why they didn't just call it an arcocele to keep things consistent, but they didn't. I don't get to name the thing. So anyway, archenteron. Another consequence of gastrulation is now we have two different components of our embryonic tissue layers. We've got an ectoderm and we've got an endoderm. So the ectoderm layer is eventually going to become things like the skin, obviously, and the exoskeleton in organisms with an exoskeleton, as we just talked about. The endoderm is going to form, among other things, the lining of the gut. All right, so we've already got quite a lot of cellular organization going on here. All right, so this is just another kind of close-up view to talk about those tissue layers. So sometimes the embryonic tissue layers are called germ layers. So if you see that term, don't be confused. These, as we said, are going to give rise to the tissues and organs in the eventual, juvenile and adult organism. We've already defined a tissue. So remember, a tissue is basically a network of cells that sort of work together to do something like, seal off a space. And then they're usually connected to some kind of basal lamina. So we're starting to form the embryonic tissues. Organs, if we want to go ahead and define those, when does something constitute an organ? That's when you've got two or more tissues working together to carry out some kind of function. So some animals as adults are only going to remain having those two tissue layers that they already have at gastrulation. These are called diploblasts. And the cnidarians are an example of diploblasts, so your jellyfish and corals and things. And then other organisms are triploblasts, and they are going to go on to develop a third embryonic tissue layer in between the ectoderm and endoderm called the mesoderm. And the way that the mesoderm forms varies among different types of animals. We're going to talk about the formation of the mesoderm next. So pretty much all of your bilateria, so all of the organisms with bilateral symmetry, which are most of the animal phyla, are going to be triploblastic. All right, so our mesoderm, the mesoderm is essentially going to be a tissue layer that's going to line the coelom in organisms that have one. The coelom is a fluid-filled body cavity. So coelom is kind of like blastocele, right? So remember that seal word means a cavity. So your coelom is a body cavity. In some of our triploblasts, though, they actually don't have any coelom, and instead, the entire space in the adult animal is actually full of cells that are derived from the embryonic mesoderm. So that's like in our flatworms, Platyhelminthes. We're going to have a cross section. This is a cross section of the organism that's going to look something like this. So you've got ectoderm that has developed into the epidermis. You've got endoderm that has developed into the lining of the gut. And then you have mesoderm, which has filled in the space in between the ectoderm and the gut entirely, that we call mesenchyme sometimes. Some animals that have a body space are considered pseudocoelomate. That just means that not all of the coelom is actually lined by mesoderm on both sides. So it's still a body space. So I don't really like the term pseudocoelomate because it's not really like a false space. There's still a space in there. It's just that what derived from the ectoderm is lined by these mesodermal cells, but then the endoderm is not lined by mesodermal cells. Okay. There aren't very many organisms that are considered pseudocoelomate. Pretty much the only phyla are the nematodes and then also the nematomorpha, which we aren't really going to talk about in this class. But there's some debate over whether this is a real thing or whether it's meaningful at all. Anyway, we're still going to learn the terms. Most animal phyla are going to be coelomate, which means that they do have this body cavity, and it's lined on both sides by mesoderm, as you can see here. And in most coelomates, that mesoderm is going to develop in the adult organism into muscles, also peritoneal tissue or parietal tissue, that is going to kind of line our organs and our body cavity. And yeah, some other things depending on the organ. A lot of organs also originate from embryonic mesoderm. All right. Questions, concerns, thoughts? Nobody? Okie dokie. This is just kind of a simplified schematic about these tissue layers. So as we said, diploblasts are going to have just the two major layers. They are usually separated by some kind of non-living jelly stuff. That's where jellyfish gets its name. But that stuff, as we'll talk about when we talk about cnidarians, is acellular. So it's just kind of jelly gunk in between the ectoderm and the endoderm. And then our triploblasts, of course, are going to have mesoderm. All right. Okay, so protostomes and deuterostomes. Have you guys heard these terms before? Yes, no, yeah. Valeria, yes. Okay, excellent. So these are kind of two developmental modes. And we used to think they were super, super meaningful as far as very deep branching in the animal family tree and animal phylogeny. However, we have more recently determined that some organisms that have what looks like deuterostomous development actually are not closely related to the other deuterostomes, which are basically us and echinoderms and hemichordates. So it's not super meaningful. Also, the more we actually study the details of animal development, early development in non-model organisms, so things that are not just like fruit flies and C. elegans and things like that, the more we learn that there's actually a lot of mess here where there's actually a mixture of these developmental types. And it's not even super well conserved within phyla. So a lot of this, take it with a grain of salt, but it's still kind of meaningful in most cases to talk about protostomus development and deuterostomus development, especially deuterostomus development, because that is pretty consistent in all the deuterostomes, as far as we know. Again, we're learning more all the time. All right. So proto, what does proto mean? Anybody have a guess? First, exactly. Thanks, Parker, in the chat. So proto means first. What do you think deutero means? Second. Yep. So deutero means second. And then what does stome mean? What is a stome, or like stomata? What is that referring to?
It's like a mouth or like an opening, right?
Yeah, like a mouth or opening. Yeah. So protostome and deuterostome have to do with when the hole that becomes the mouth develops during gastrulation. All right. So in our protostomes, the blastopore, that first hole that leads into the archenteron during gastrulation, is going to become the mouth. So that's in protostomes. In chordates like us and echinoderms, and hemichordates too, the blastopore is eventually going to be what becomes the anus. And later there's going to be another hole, but that's a bit later in development, that is going to be the mouth, and the opposite is true in protostomes that have a complete gut. So later, if they're going to have an anus, and not all the protostomes do, If they're going to have an anus, then another hole is going to form to create that tube within a tube structure that we see in adults that have a complete digestive system. All right, so that's where the names come from. These things also differ in how their coelom forms and essentially how their mesoderm forms. So if we're talking about a triploblast, our protostomes are going to have this sort of solid mass of cells kind of on either side of the gastrula or of the archenteron towards the blastopore, but sort of in between the ectoderm and endoderm. So these cells are going to form and they're going to, eventually sort of fill up this space. And the space in between those cells there is going to hollow out, and that hollow area is going to become the coelom. Okay. However, in the deuterostomes, what tends to happen, and this is where you really start getting messed. This is not what always happens. What tends to happen is called enterocoely. And in enterocoely, that mesoderm, rather than just forming as these separate clumps of cells, you actually have an invagination of the the endoderm, sorry, that is eventually going to expand to line the ectoderm and the endoderm, and then the spaces here are going to sort of become the coelom. So that's called enterocoely. This version that we see in protostomes with the clumps of cells is called schizocoely. There's also differences in the pattern of cleavage when the cells are getting into that 8-cell stage, right? So we've got, 4 cells on the top and 4 cells on the bottom. And during the cleavage, when the cells divide to reach that 8-cell stage, in the protostomes, there's going to be this like twisting. So you can see that, like, for lack of a better word, the crack in between these 2 cells on the bottom is not lined up with the crack between the 2 cells on the top. It's kind of twisted. Whereas in the deuterostome, the cells kind of line up with each other. So there's not this twisting. So that's called spiral cleavage in the protostomes. And in the deuterostomes, where everything stays lined up, we call that radial cleavage. So radial is like rays. So I don't know, I imagine like little sun rays coming out of where the blastopore is, and they're sort of in a line, right? So the cracks are lined up. That's how I remember it. This is how I remember stuff, by the way, and this is how I remember all this junk when I was an undergrad too. Like, you make up little mnemonic devices or like whatever you need to like remember it. So if you need to remember a little sunshine shining from the blastopore with rays going around to remember radial cleavage in deuterostomes, then yeah, by all means. All right, so again, not all of the protostomes that have now been studied do have this spiral cleavage. The other difference is in whether or not the cells, and actually I don't have this on here, whether or not the cells at this stage are sort of predestined in terms of what they're going to turn into. So at this super early stage, each of these 8 cells in a protostome already has essentially determined like what it's going to turn into in the adult. That's determinant. Okay, determinate development. Indeterminate means that these cells still have what's called totipotency, so they could turn into anything. So at this stage, deuterostomes still have indeterminate development. The blastopore we already talked about, and I just mentioned that first because that's where that term protostome and deuterostome come from. All right. Excellent. All right, and so this is just another like simplified graphic that shows the differences between schizocele and protostomes and enterocele and deuterostomes. Again, you have to qualify that with most on both of those. And this is just another little diagram that gives you some more detail about the cell cleavage patterns when we're going from 4 cells to 8 cells again. And I like this because it really looks down on the cells from the top and bottom. And again, you can see that when you have radial cleavage in your deuterostomes, you've got the sun shining out of the blastopore. And I don't know, isn't this, wait, is this a thing? I said the sun shining out of the blastopore, but if you really need to remember that it's the deuterostomes that have radial cleavage, you can picture the sun shining out of the anus. That's the thing, right? Because that first hole, the blastopore, is going to be the anus. Anyway, so I don't know. Have you ever heard somebody say like that he thinks the sun shines out of his ass or something? Anyway, that's how I remember this stuff, guys. So yeah. So sun rays, radial cleavage, deuterostomes. All right. Okay. So we mentioned that some of these things, these characteristics we talked about so far are in the bilateria. So remember, all the bilateria are going to be triploblasts, right? So the cnidaria are diploblasts. So they've only got an ectoderm and endoderm. Whereas the bilateria are going to go on to develop that mesoderm. And then in terms of deuterostomus or protostomus development, our deuterostomes are going to tend to have that deuterostomus development, hence the term. And these other guys in the ecdysozoa and spiralia are going to tend to have the protostomus development. Okay. All right, so now we've talked about all those things. Any terms that, or any questions about, before I move to the next thing, any questions about development? Embryonic development is something where I find students sometimes struggle in exams. So make sure if you have questions about that, or you want me to go over something again, go ahead and ask. And also make sure you study that part really carefully. No questions? Okie doke. All right, so we're going to talk now a little bit about body plan development. So like I said, we're not going to talk a whole ton about this because, yeah, this is just a whole other course talking about transcription factors and body planning genes. Oops. All right. So animals have pretty conserved body plans, right? And it all comes from these conserved genes that determine how our bodies are going to be shaped, essentially. So most of the multicellular eukaryotic lineages, right? So that's like fungi and animalia, or metazoa. And, you know, there are other little minor relatives that we talked a little bit last time about. Most of them are actually going to lack a specific body plan. This also goes for the plants, right? So, like a seaweed can have a thallus, which is basically their body, that really varies a lot between individuals. You know, cells don't have as specific a fate. But our metazoan eukaryotes are going to have a more conserved body plan. So, like your tetrapods always have the same number of appendages, right? So we all have 4 appendages. You're never gonna have, and this is why dragons can't have existed, right? Because they've got like an extra set of appendages. That's just not a thing, because of these really, really, really conserved body planning genes that don't really change much. So yeah. Also, you're never gonna have turtles with wings, or yeah, stuff like that. Okay. So body plans in different groups of animals can do vary a bit. So sponges have an asymmetrical body plan. Um, Cnidarians and also placozoans have this radial symmetry. And then secondarily, actually, echinoderms have radial symmetry as adults, but we'll talk more about what's up with that later. And then we've got what's called biradial symmetry in ctenophores. We'll talk about what that means when we get to ctenophores more. And then bilateral symmetry is what we have in all of the bilaterians, which is pretty much everybody else. All right, so what do those things mean really quickly? Asymmetry, again, that prefix a means without or no. So sponges are going to be asymmetrical. Their cells can be grown every which way. Some types of sponges are going to look quite symmetrical, but if you look at really like the cellular structure and body structure within, you can still see that asymmetry. Radial symmetry basically means that you've got like sun rays shooting out of a central you know, area. So just like radial cleavage in our deuterostomes. But radial symmetry, another way of thinking of it is like, can I cut this thing like a pizza and end up with more than 2 equal parts? So if you take a sea anemone, which is an cnidarian, you can cut it like a pizza and you end up with equal parts, you know, with some tentacles and some gut and other stuff and muscles and stuff in each one of those pieces. And again, the same thing with echinoderms, like the sea star here. But that sort of evolved a second time separately in them. And we'll talk much more about that when we get to echinoderms. Bilateral symmetry. So bi means 2, lateral means sides. So bilateral symmetry means we have 2 sides. So we have essentially a left side and a right side. Also, some other interesting things happen when you have a left side and a right side. When you have a left and a right, you also tend to have a dorsal and a ventral, or kind of a top and bottom. You also have a front and a back, so an anterior and posterior. And And this is really, and you'll see when you watch, if you haven't yet, that video that is linked in your textbook on the origin of animals. It talks quite a bit, I believe at least, or we'll talk more about it later when we get to just talking about bilateria. Having a head end um, ends up giving you a lot, right? So now you have a direction that your body's going to move in. You can have cephalization, which is essentially having a brain and sensory structures concentrated in that anterior region, and a bunch of other stuff. So anyway, that's bilateral symmetry. Um, Yeah, so I got ahead of myself a little. So yeah, cephalization, having a head area, you can have that when you're bilaterally symmetrical. When you have radial symmetry or asymmetry, you can't really have a head. Instead, as we'll talk about with cnidarians, what we really have is an aboral side and an oral side. So which side is your mouth on? Which side is the opposite of that? Rather than really having an anterior and posterior. All right. So I want to end with a question. So anybody know what this is, first of all? Yeah, Parker said in the chat it's a brain coral. Yeah, it's a brain coral. Okay, so looking at this thing, what kind of symmetry would you say that this organism has? Okay. I'm seeing Parker is saying radial symmetry. So why are you saying radial symmetry, Parker? Can you kind of justify that? Alex said I was going to guess asymmetric. Yeah, so Parker said it's roughly circular. Alex, what were you going to say asymmetrical?
I feel like it is roughly circular, but it's not like perfect. And like looking at like the edges of it, like if I was to like cut it like a pizza, like what you said, like it wouldn't be equal. Like
It wouldn't be equal.
it would have different types of crust.
Right, yeah. Okay, so this is kind of a trick question, and the reason I asked you guys this way is because you really have to think about like what does it mean? What is the unit, right, of the organism? Um, a coral is actually a colonial organism, right? So it's made up of a bunch of little structures called polyps. So if we zoom in, we've got all these little individual polyps in a coral. Now they are clones of each other, that form by basically dividing in half by fission. The original polyp was a product of sexual reproduction, though. So they do have sexual reproduction. But that polyp is going to divide and divide and divide, and then we end up with a whole colony of individuals. And so this kind of macroscopic structure that you look at when you look at something, and you would call that organism a brain coral, is really made up of a colony of individual polyps. And the individual polyps Guess what they look like? They look like little miniature sea anemones. So each little polyp making up a coral is like this, just like a sea anemone. And each one of those polyps is what kind of symmetry? What would you guys say the symmetry of the individual polyp is?
Radial.
Radial, exactly. And so, because the individuals are actually the polyps, we would still consider a coral like this, or even like a branching coral. Maybe I should change this slide. If we had like a branching coral, like an elkhorn coral or something like that, where like it's really clearly like macroscopically asymmetrical, you would still say that corals have radial symmetry because the polyps have radial symmetry. Does that make sense to everybody? Yes. Okay. All right, so we're gonna pause here. We're gonna take about a 10-minute break, and then we will come back together and we will start the next lecture on the chordates.